पूजा आपकी स्टार्ट।
So we are starting our lecture 6 at 12:45 p.m. Molecular biology of carcinogenesis and metastasis related lecture by Professor Neharika Nath, Madam. Madam, please you can continue.
Thank you, Pooja. Welcome again. I don't know why my screen is coming. Let me stop share for a bit. Welcome to part 2. And today we will talk about the basics, the real basics, which took us very, very long to do, and that is pure molecular biology. So
recording in progress.
Molecular biology refers to the cell at the unit level, then anything inside the cell, the proteins, the sugars, everything at that molecular level. So molecular biology has a lot of names. So inside the cell, if there are so many proteins and sugars, then every protein and every sugar has a pet name. And that's the way it's studied. So you also have to get kind of like used to certain names. And I've been giving you a couple of names for proteins because they're all like individual people. So in a house, you have people living in a house and everybody doing their own work, hopefully, their own share of work. Everybody has a role. So same way, the cell and whatever it's doing has all of these proteins. Mostly proteins are the workhorses. So everybody has a name. So every protein will have a name. So you'll see a lot of names today. If they ring a bell, good. Like if you remember them or if you've heard of them, good. If you can't remember every one of them, you get the general picture. And after your workshop, there will be a little quiz of a couple of questions, quiz or test, I don't know what to call it. And so the general questions will be there. It will not be very detailed in every protein, but you should be able to name some of the famous ones that we will talk about. So please pay attention today. So molecular biology of whatever we've done so far, the molecular biology of that. And you've already heard a couple of the names. So let's go dive into it. So first of all, you heard that the risk factors, there are risk factors, and we talked about that essentially it's a genetic disease. Cancer is a genetic disease. Oops. And so those cancers that are there with inherited mutations, I thought, why have every mutation up here, but what about the only ones that are really inherited? So if your mother or father has those mutations, or it runs in the family, then it's good to know as a member of the family whether you do or you don't have it, and to be careful and have that guidance. And so we found that certain mutations get carried on. So this is a graph of the percentage of the tumors, all those tumors of all of them, all the different cancers, those with inherited mutations. So the mutations will be passed on from mother to daughter. And so when I say mutations, that some of those defects, whichever they are going to be passed on. So and it's not possible for everyone to know every mutation that's here. But in general, you have an idea. And so, ovarian cancers come with this inheritable mutation, and that's about 19%. Stomach, there are some, I'll give you one or two names. Stomach, there are some genes, and colon and rectal, there are some genes that are that runs in the family. Breast cancer, the very famous is BRCA1, BRCA2 gene. If that's a mutation, then from mother to daughter, then it very likelihood that the daughter should start observing, should get herself tested and be alert from a very young age. And again, it's not all breast type of breast cancer, only certain type of breast cancers. The less aggressive, the more aggressive breast cancer is not in this category. Um, let's see. Yeah. So, prostate cancer, lung cancer, certain mutations, only up to 8%. It's not so much. So, as you see here, even up to in blood cancer, AML is at the bottom. Brain cancer, glioblastoma, certain mutations that can be inherited from a parent to child. Okay. And so the lung cancers, there are different types of lung cancer, small cell, non-small cell lung cancer, NSCLC. So there's different types of cancers. That's why it's written as like lung 1 and 2. Okay. So let's just look at um um in cancer risk, we said that again genetic is a factor. So again, one more time, mutations. And so this is a fairly old graph, but it is just showing you that if if there is a mutation, then the chances of developing the cancer compared to the general population, goes up a lot. Okay, this is just to show you that um, if your parent or someone ran in the family, then having that mutation increases, or having another mutation increases a lot. So here we have just a couple like breast cancer and colon cancer. Let's talk about that. So in um, in colorectal cancer, as you see, by age 70, if the general population just has a 2% chance, but then if an individual has the HNPCC, so if you see at the bottom, if an individual has HNPCC gene, then um, uh, then HNPCC, we call it as gene, but it's a condition, the hereditary non-polyposis colorectal cancer. Then that genetic disposition means that you will get the Lynch syndrome, and it can lead to the colorectal cancer. So these are different kinds of mutations, but it shows you that the chances go up by 82%. So it's like how bad is it, and when will that person get it? So in breast cancer, it says here by age 50, if there is that BRCA1 mutation, and um, those who get it by age 70, because of age, there is the tendency to then that the cancer will show up. So a lot of the signs show up all between, early 40, even 47, 45 onwards, all the way up to 70. So what is the risk? So if you look at it, breast cancer has one of the highest risk, 87, colorectal cancer, high risk of 82, and the other cancers, ovarian and endometrial, follow suit. So having that mutation, having the mutation increases the risk a lot. Oh, and I forgot the spelling of cancer. There you go. Alrighty. So now again, you cannot escape those hallmarks of cancer that we keep on saying. So everything kind of like revolves around that. Hallmark of cancer is capability. What is that identifying feature? Okay, it's like feature, it's an identifying feature, hallmark of cancer. And so we said that they were initially six and then there were some more. So I just have seven here. And so everything that happens is around this. The capability of the cell is around this. The capability of the cell, you know, works because of these properties, because of these features, and it acquires properties because of these features. So, let's see. So I have here a big list for you now. Molecular alterations that drive these tumor initiation, progression, and metastasis. Remember, three stages: initiation, promotion, and progression, and then metastasis. So total of four, or you can just say three, and compile one. So what are the molecular alterations? And this is really a big summary if you ever needed to go back and look at it. So it's just a big summary. And so there's like 2 pages of this. It's 2 pages. So it's kind of really fairly huge, and some of them are new. So up in the beginning, not like I have to read all of them, but up in the beginning, if you look at it, there's that gain of function, those famous words that you hear, gain of function of a oncogene. Okay, oncogene that gets activated. Oncogenes are those are the names: RASC, MYC, EGFR, that's epidermal growth factor. This is a growth factor. That's the receptor, EGFR receptor. So this is an oncogene, which when it says gain of function, that simply means that the a small one mutation or less mutations give rise to more activity, which is really bad. Okay, so if you have a growth factor and there's a mutation, then it immediately that means it makes proteins that keep on working. They keep on. It's gain of function. The mutation just gives you more function. Instead of not working, it works too much. So tumor suppressor gene, on the other hand, if it is mutated, and your favorite ones that you're going to look at today, and I'm also going to give you I think I might have a video, is P53 and RB. These are tumor suppressor genes, famous ones, and their job is to, if the cell is not doing too good, very abnormal, go and the cell will have to die. So they will stop the cell from multiplying. They will keep it quiet. And so the good thing about this one is that if this has a mutation, P53, if it has a mutation, then there is it's a loss of function. It doesn't work, but you need on both chromosomes, on both alleles, you need to have the mutation. Then it will not work. Okay, so that's the good thing about it. Whereas in that oncogene activation, just any one allele, one chromosome has a mutation, it will overdrive. It's overactive, which you don't want. So tumor suppressors prevent too much cell division. Oncogenes promote cell division. All right, so that's the basic stuff. And then there's this famous one. So we'll look at some of these, though important ones. We'll look at telomeres. Epigenetics is something that's fairly new. Epigenetics means not very new, but 20 years new. Or 20, 30 years new, let's say. So epigenetics is where besides the mutation was not there. There is no mutation, but something happened to the DNA where it got a methyl group added. Okay, it had a methyl group added to it, and the DNA became silenced. Okay, so led to gene silencing. Or the chromatin on the DNA was modified. Here's the chromatin on the DNA. It got modified and the gene expression changed. And the gene is there. It's not mutated, but it just doesn't express, or expresses, or it doesn't express. Okay. It doesn't do its job. Then, so this is fairly big up here, but things that you recognize: angiogenic signaling. This is a summary. We haven't even started yet. This is angiogenic signaling, so like vascular endothelial growth factor, VEGF. I think maybe we'll talk about it more today. So some of the things that you already know, Warburg effect. Yesterday I talked about the HIF factor, HIF, the HIF gene. And so then for EMT, today we just talked about EMT, and they have very special genes that do that. But then if E cadherin is down regulated, then it'll become mesenchymal and it'll promote the tumor. So let's look at something that is general and not extremely new. So we'll look at a couple of examples. So here's one that is kind of like fairly new, and this is like microRNA. MicroRNAs are fairly new. Okay, the non-coding RNAs, L long coding but long. Non-coding, they don't really code for a protein, but they have regulation. They control things. So LNC, LNC RNA. These are long coding RNAs. These are fairly new for the last several years, and they can also, um, if they're present, then they can also push proliferation and push cancer. So with this, let's move on and just let's look at some the molecular biology of it. And so, to explain these molecules and to explain these names instead of just reading out things from a chart, the best way to learn these and to understand these is through little experimentation. So if you have normal cells and you grow them in a growth media, yesterday I showed you a flask, then you give it this growth factor called as platelet-derived growth factor, then the cells, they will multiply. Okay, then they will multiply. And but in cancer cells, even if you don't give in cancer cells, even if you don't give any of that PDGF, that signal, then still they keep on multiplying. All right, so what would happen? Normal cells, most normal cells, they divide with PDGF. All right, and cancer cells, many cancer cells, not all of them, they will just grow in basic growth medium without, without the signal. Okay, without the signal. It's like they have a mutation. They don't need a signal to do anything. All right, they don't need that signal. So it's like your mother gives you food at dinner time, but if she didn't give it to you and you were really hungry, you're going to just go get it yourself, so that's the kind of thing. You don't need mother as the signal, you don't need the signal, you're just going to go and get it. It's that way. So the other thing about these, so PDGF, so growth factors. So what do you need? So you need, so essentially you need basic growth factor signaling. And in cancer cells, you don't need the growth factors signaling, or it is signaling by itself and it's doing too much. Okay, so and this is just a property that these cells, the normal cells, will grow to a certain extent, but cancer cells will grow on top of each other, will grow on top of each other. So they have certain genes that are telling them not to be contact inhibited. They just don't. They don't bother for contact inhibition, and so there are certain genes that will tell it you can keep on growing. You don't have to have a nice layer. Just keep on growing on top of each other. Um, so in terms of molecular biology, there are signals that that allow it to be. Um, there are adhesion molecules that it is modifying. All those adhesion molecules, it's modifying them, and it is saying, okay, we can have a bigger crowd of cells. Okay, so they either they will create the adhesion molecules or they will break them as necessary. So that's your adhesion molecules. Then they have cancer cells just they don't respond to apoptosis signals, so they will not listen. But if there are certain special signals, if there are special signals, they will try to evade it, and so they have a mechanism. So they have a mechanism to evade it. So here it is. So normal cells will Normal cells will undergo with have certain signals that are pro-apoptotic. Okay, normal cells will respond to pro-apoptotic proteins. Okay, I'm just calling it proteins. And cancer cells, they will get they will get the pro-apoptotic signals, but they will make anti-apoptotic signals. And believe it or not, they do exist. Anti-apoptotic proteins. So the cell was making, when it's needed, it will make pro-apoptotic proteins. Pro means apoptosis is cell death. It will go undergo apoptosis if there's a problem. But cancer cells, number one is they have a mutation and they figured out how to make anti-apoptotic proteins, so they just don't can ignore the pro-apoptotic proteins. So that's what they will do. So in terms of their biology, they will make in anti-apoptotic proteins. And there are certain names to this, and I don't want to give the name right now, maybe later. And then the molecular biology of these cells is such that they need to, normal cells, they need to, as they multiply, they in their chromosomes, these normal cells, as they keep on replicating, the normal cells, they get short. So every time your cells are replicating over the last 25 years, let's say, or how old you are, your ends of your chromosomes have these telomeres. So this is a chromosome of the adult cell. And the end of the telomere is getting, is just not there anymore. Over time, it doesn't get chewed off, it's just not made, that's all. So it just gets shorter and shorter. So telomeres, oh sorry, telomere is that little edge here, that little end of the chromosome. And I'll not go into why we need those, but we need those. Um, so those telomeres over time, over your 25 years or over 40, 50 years, they are getting they are getting short. So look at this telomere, it's short versus this one. This was fairly big. So they are getting removed. Okay, they're just not being made, if that makes sense. So the telomere shortens. So short telomeres. So shortened telomeres. Telomere is nothing but whatever's at the tail end. Telo and mere. So tail end units. And so the telomere is now short. And that becomes a problem. Telomeres that are short, that are too short and very close to your original chromosome, very close to your original chromosomes. Here's where you have all your genes for all the work. Okay. However, telomere don't have any genes for work. Whatever you're doing through the whole day, the telomeres don't have any genes there. They're not doing anything for you except for except for this work of um, keeping you young, in fact. So as you age, that telomere becomes short until it's not there. And in and that's like a normal thing. And so therefore at a certain time the cells are at senescence. Senescence means old age, and they know that now is the time just to maintain and not to multiply too much. And so that's what means for telomeres at senescence. So senescence means old age of the cell. And in cancer cells, the telomeres, if the telomeres were short in cancer cells to begin with, let's say at the age of 50, like you just saw, 70, right? The telomeres are already short because the person has already lived 70 years of age. And now there could be certain changes where the cell decides, okay, I want to add the telomerase enzyme. Let me start making the telomerase enzyme and let me add some telomeres and let me make my telomeres big like before. Okay? Let me add in telomeres before. And so uh, this this chromosome now behaves like something like this. It just thinks that it wants to multiply. And so having long telomeres kind of like is also a sign that it just needs to multiply a little irregularly or unnecessarily. That's the word. So telomerase enzyme, by some kind of mutation, is just made, and if it's made more, then that becomes a problem. So we have a problem there. Okay. And so that's so what is the molecular biology of this? The limitation on proliferation that was there, okay, that is overcome. All right, it just bypasses it, overrides it. That's the right word. Let's say. Um, okay, so I had some, I think that's a lot of details for telomerase and telomeres, so we don't need to know this, but, and the method by which this occurs, so I would just let it go. But the bottom line is the enzyme that makes the telomeres, that enzyme that makes the telomeres got activated. And so the next is a major molecular biology, is the cell cycle. And in the cell cycle is it's like a clock. It's like a clock of the cell that wants to divide in a timely manner and not over divide and have a lot of checkpoints. So this is a clock. This is a clock, and it's a representation of the cell that follows this cell cycle. And in this clock, the a new cell, which has just been two new cells that have just been made, both of them, like in your GI tract, in your stomach lining. Remember, I said every 4 or 5 days cells have to be made. So let's say this is the GI tract, and so let's say you have a couple of cells here, but this is one cell. And so this cell needs to, it in the clock for a good time, let's say um, for about 12 hours. Okay. Oh, why is my sharing paused? What happened? It says my sharing is paused. Are we okay? Can you hear me, anybody?
Yes, ma'am.
Okay.
We can hear you.
Yeah, it just said my video was paused for a minute. I mean, my sharing was paused. Okay, so I just reshared. Oh, let me put it back in. Yeah. So for a good, well, so this is the cell cycle. This cell cycle is like the most critical thing for us. This is, I live and I breathe the cell cycle every day in my research and everything. All right, so the cell cycle is made up of all these phases. There are a bunch of phases, and so G1 phase, the S phase, the G2 phase, and the M phase. So these are the four phases of the cell cycle. Okay, and this is like a clock. So for about 12 hours, this new cell of your stomach lining or your GI tract, this for about 12 hours, it will simply grow a little bit big. It will make more proteins. It will make more it will just go in size and do what it has to do, but it's not necessarily working right now. It's just a newly born cell, right? It's been divided, and it hasn't started really working yet, but it'll spend those 12 hours a little bit of growing, okay? But it's not really actively doing any active work. Like, it's not making any enzymes. It's not really mature that much yet. Okay, and you can ignore this as G0 for now. So after 12 hours, there is a checkpoint. There is a, after this 12 hours, here we go. So I'm here right now. There is a, around this 10 to 12, 10 hours, there's this checkpoint. And at this time, the cell really checks everything. Do I have all the proteins that I need, etc., blah, 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 and all that stuff? Do I have everything that I need? Is there nutrition? And then this cell would like to replicate its DNA. All right, so it's decided that it's going to replicate, and it's going to make some more cells because the GI tract needs it. So it's going to make 3, 4 cells today. So then the DNA synthesis takes place. And the DNA synthesis, so for example, you have 23 pairs of chromosomes, right, as humans. And so the DNA will divide and it will multiply and it will make about 46 chromosomes. Okay, so it'll The chromosomes are not there yet, but the DNA amount is there. So it's like it's it's yeah, just for regular purposes, just say that it's making double the amount of DNA. So it has committed. So from this checkpoint, it is committed. Then the next job it will be is to get ready for another round of division. And so it's looking for, do I have my chromosomes in place and everything? And so there's another checkpoint here. So that's called as the G2 checkpoint. And as all the chromosomes and everything is there. Do we have enough energy? Do I have enough nutrients? Can I make in two cells? And here's when the cell divides into two cells. So this one here divided into now two cells, and that's kind of like a short phase. So it'll go for 12 hours, we're just preparing, and then maybe for the next, um, I don't know, 6 hours, maybe it will do the S phase. Then for about 4 hours, it'll do the G2 phase, and the M phase will, the mitosis phase, cell division, will be, let's say, 2 hours. Anyway, so it'll kind of like make up about 20 to 24 hours. The total cell cycle is 20 to 24 hours. So if I have one cell today, it'll take 24 hours for it to grow and to divide into two. Okay. So if it, I don't know if it's 10, 20, yes, this is 24 hours. So it's about 24 hours. So there's also another checkpoint here. So that's called as the metaphase checkpoint. And are the chromosomes, are all equally going? Let's say sometimes you have Down syndrome, you have this syndrome and all that. That's where at the metaphase, this checkpoint, it failed. And there was either one extra chromosome here or one less chromosome there, and that can become a big problem if that happens in the sperm and the ova, um, and that gives rise to a Down syndrome baby, let's say. So in cancer, these checkpoints are not going to function. The cancer cell will trick it and just say, oh, there's not enough nutrients. Keep going, keep going, keep going. All right, it ignores the checkpoint. So these are like gates, this checkpoint. It ignores the gate. Something wrong? DNA is not replicated. Fine, fine, no problem. Keep going. All right. So the checkpoints are ignored in in cancer. So any one checkpoint can be ignored. And if there's a DNA repair needs to take place, it will not happen. But normally what happens is that, let's say there was UV radiation and there's some cells that have a damage. Normally what would happen is it would go and stop here and the cell would wait and then it would fix the problem. It would add the DNA repair enzymes. It would fix the problem and then it would start. So it would wait and then it would cross the checkpoint. So at the gate, it would fix it. So if you went to the cinema and you forgot your tickets, you have to go back home, get your tickets, and then you're allowed in the movie hall, right? It's the same thing. Okay. So so this is kind of like whatever I said, it kind of like explains the same thing in the same manner. But the bottom line is that these checkpoints, um, if there's any instability, then these checkpoints are kind of ignored. If there is a mutation, then the checkpoints are ignored. So if there is a P53 mutation, okay, then and if there's a RB mutation, then the cell, whether it's good, bad, whether there is DNA damage, it don't care. It just keeps going. Okay, so P53 will tell the cells to die. It will not die. So then it just doesn't listen. Okay, the cell just doesn't listen. If there's a DNA has not replicated, doesn't matter. Just keep on multiplying, and so two cells will become four. Four cells will become, 8, and it'll keep multiplying and be useless bunch of cells. So genetic instability. So I said the words proto-oncogenes and tumor suppressors, and so these are very, very critical types of cancer genes. The very critical types of cancer genes are proto-oncogenes and tumor suppressor genes and DNA repair genes. So let's just talk about the proto-oncogenes. So I told you that proto-oncogenes or oncogenes, we use the word, we call it proto because it's just before it can become an oncogene, so or can lead to. So a proto-oncogene is a gene, and I don't need to name it here, but it's a gene that promotes the cell cycle. So this cell cycle that you had, its job is to tell it to go, keep going. Not keep going like crazy, but to keep going. And so we have normal proto-oncogenes and they keep working. So they're the ones who tell the cell to cycle. All right, one cell divide to two and follow the protocol. But here, these proto-oncogenes, they promote the cell cycle. But if there is a mutation, then they gain function, like I said. And if there's a small mutation, they will suddenly go to overdrive and they'll just say, okay, everybody, keep on going. Keep on going. So it's like constantly telling everybody, instead of being a nice gatekeeper, everybody keep on going, everybody keep on going. And so oncogenes, when it is mutated, when it is a gain of function, it is called as a oncogene. It is called as a oncogene. So oncogenes result in hyperactive, too much proteins. Okay, so what does that mean? If you have a car, then you just have, it's going this way, the car is going this way. You have too much gas. Gas in America is really petrol. So we're just saying it has too much petrol, and it just, it's like it just cannot stop. It doesn't know how to stop. It just keeps going. All right, it's very sensitive, just keeps on going. Okay. So the next one is tumor suppressor genes, TS, and that's your RB and P53. And the example for this one is that uh, if the tumor suppressor gene, this is the brakes of the tumor suppressor. This is the brakes of the tumor suppressor. This is the tumor suppressor. It's the brakes, and it just doesn't work. Then the car does not stop and keep going forward. So if there's a loss of function and if there's too much of loss of function in both chromosomes, then it just goes ahead and keeps on multiplying. So you cannot hold the horse back; it'll just keeps going. So two copies um, have to be loss. Loss means it's either a defect or it's just not there. The gene for P53 and what did I say? What's the other one? Um, I forgot. RB. Yes. Anyway, so the next is DNA repair genes. DNA repair genes, they will work. They work all the time actually, and so DNA repair genes, if there's any defect in a DNA repair gene, then if there's a break, look at this. There's a break, then it just does not repair it, and it can make mistakes, and then it continues. So they keep the genome unstable. And so how does this proto-oncogene become an oncogene? Whatever happened? Well, it can happen in various ways, and I think that's fairly a large amount of details to go through, but I will just make it short. It can. So here's your gene, just so that you have an idea also. So this is your normal proto-oncogene. So this is your DNA. You can see this is the DNA strands, and that's your gene, whichever the gene is. Okay. So that's your gene. And so if you have too much of proto-oncogene, that's cancer. Because what is a proto-oncogene? It it tells the cells to divide. It forces the cell cycle. It tells the cell to divide. How does it do that? It tells the cell cycle to continue. It tells the cell cycle to start, at least. Tells the cell cycle to start. So if you have this gene and it makes this gene, so this is the gene here, and it makes protein. So genes, they transcribe, they translate, and it makes the protein. So this is your proto-oncogene protein. It's the same one. This is the gene and it just makes it. Okay, so it's the same. This is the gene that makes the corresponding protein. And so it's possible that under normal circumstances, look at this, there was nothing else here. So before the gene, there was nothing else. But in this case, in this example, there is a addition called the promoter, and the promoter just tells the gene to keep on making protein, and then you have too much of proto-oncogene, and then you have too much of cell cycle. So again, you have too much of cell cycle. Cell cycle is just what I showed you before. It's like a clock for the cell. So you have to, what you needed was just one protein. You know, here, just like one, that's how much you needed. One gene, one protein, but you're making three here. Next situation, so this is called as the gene is translocated, okay, and placed under a new promoter. Next thing that can happen is that the gene gets duplicated. You have a gene here, you have a gene here, and you have another gene. All right. Now that's all okay for if you had if you had some kind of it was not harmful. That's okay for a gene that's not harmful. But for a gene like a proto-oncogene, which tells the cells to divide, that start the cell cycle, if again you have too much of genes and you make too much of the protein, and when the gene is duplicated, you just make too much of excess protein, again you have too much of cell cycle. Same. Okay. So too much cell cycle, too much cell division. Same thing. Then next one is a point mutation. If instead of the full gene you had a break in some gene, it's a point mutation, then also it's possible that it makes some of the point mutations lead to more protein, because this mutation would have given you a little bit of protein. I mean, normally it would have given you a little bit protein, but this mutation here just changes it so that it makes more protein. Another problem. Okay, so problem galore, right? Any small problem with proto-oncogene becomes a big problem. You just make too much protein. And sometimes there is a point mutation right here in the gene, and it just makes this point mutation. So there's a mutation right here, or small mutation here, and normally you make one protein and it functions, but now it's this one protein was either active or not active. But in this situation, it makes protein that's always active. Too much work. One protein constantly working does not take rest, and so that leads to a always active protein. So it becomes always active protein. You don't want proteins to be always active. So, so that becomes an issue. So next is the tumor suppressor. Now the tumor suppressor, we're not going to go into the full details of this, but a tumor suppressor gene is supposed to halt. And if it doesn't work properly, okay, this is just too much to talk about today, but it's there. Um, there is if it doesn't work, so this is the example I can give you. If this tumor suppressor gene, if it's deleted, it's gone, and this chromosome now is just does not have it. Look at this here. Then that means that's a big problem. That means uh, the cell will not stop, okay, because its job is to halt the cell cycle, okay? So they halt the cell cycle. The cell cycle needs to take some rest, right? So they halt the cell cycle because the cell cycle needs to think, decide what to do, what not to do. So generally they halt the cell cycle here. The stop sign is just not there. So one tumor suppressor is P53. And so P53 is called as the master regulator, okay? It's called as the master regulator. Why? Because it leads, if there is any, it's a master regulator because if there's too much signal from somewhere else, too much growth factor, it's from another cell, too much damage, DNA damage, all right? Or the telomeres just got short, all right, and just got too short, not good. Or if there was low oxygen, remember, so cancer cells are in the center of cancer cells is low oxygen. If there's normally low oxygen, this P53 is going to get active, and look at this beautiful sunshine. It's going to get active. It's like it wakes up, and then it will cause the cell cycles to arrest. The job is the cell cycle needs to stop. Don't multiply, just stop. That's a good thing. So it will cause arrest of the cell cycle. It will not go to G1, S, G2. It'll not continue. It'll stop and it'll try to repair. It can cause senescence. Senescence is old age. It can tell the cell, hey, keep quiet, don't do any work, just sit there, do nothing. All right, and but don't multiply. And if necessary, it can cause apoptosis. So if p53 can lead the cell to death when it cannot fix. When can it lead the cells to death? When it could not fix the problem. The cells were arrested, it could not fix the problem. P53 will just say DNA is too bad, let's just kill the cells. And so if p53 is mutated, we have a big problem. If this p53 is mutated, then none of the, it will, nobody's here to stop anything. DNA damage, who cares? Telomere shortening, who cares? All right. Um, there will be no cell cycle arrest. This will just not happen. Okay. So, oh, too much signals, who cares? You know, so there will be no cell cycle arrest, and there will be no apoptosis, there will be no senescence if p53 is mutated. So if it is mutated, then it just doesn't work. See that sunshine? That sunshine is just not there anymore. It's just a boring protein that refuses to work. And so if cell cycle does not arrest, does not take place, and that's a big, big problem. Okay, so long story short, if there is p53, that's your master regulator. All right, it's the big boss. And there's any DNA damage. How do you have DNA damage? UV light, too much sun, etc., etc. You know, so gamma radiation, stuff like that, they damage the DNA. Okay, so gamma radiation, UV radiation, you know, which you're getting by exposure to sun, they p53 gets activated, and you don't need to know all these proteins, but it causes cell cycle arrest, or it'll cell cycle death. And then the cells will, you know, they'll wait. They'll think about it. But if p53, so if p53 doesn't work, then there's no cell cycle arrest. And let me just take you quickly back. And then the cells, they don't stop. If they don't stop, they're supposed to stop here and wait. If they're not going to stop, they're just going to go, they'll replicate the DNA. All right, DNA is damaged, they'll replicate damaged DNA. Things are not good. All right, DNA is damaged, they'll still get ready to divide. DNA is damaged, they'll keep on dividing. Okay, no stopping it. And so about 30%, no, about 50% of all cancers, 50% of all cancers have p53 mutations. Sorry for the bad handwriting, my pen is crooked. So about 50% of all cancers that you see, they have p53 mutation. And so that's why it's the master regulator. So another one, same thing. When you want to shut down, so there's another one called as p21. It's on chromosome 6. It also causes cell cycle arrest when things are going wrong. Now, it's like the it's like the worker for p53. P53 wakes up, look at the sunshine, and it tells p21. So it makes p21, and p21 is this. This is p21. See this hugging protein? This is p21. And so P21, all P53 is the master. P53 is the master. It says, hey, P21, go and do the job. And so then P21 is the worker. P21 goes and stops the cell cycle. You don't have to look at this here, but it's good to know that P21 just hugs everybody and just say, okay, okay, don't do any work. Gives a big tight hug, and that way p21 is a inhibitor, and it inhibits the cell cycle, which is a good thing when things are not good. Okay, when things are DNA's damage. So this is just a short question for you. I thought I would put in and make you answer. So if both p53 alleles on both chromosomes, if they were non-functional, then what will happen? Then there's a big problem. That's what will happen. Okay. They will just not make, p53. Remember, I told you that p53 is known to make the p21 protein, so it will not make its dedicated follower p21. It will not be made, and if p21 will not be made, then obviously the DNA damage will not be repaired. and the cell will just keep dividing. Okay. And same thing with the retinoblastoma protein. RB protein is also a tumor suppressor. It goes and binds to the DNA and it gives it a big hug so that this gene here, this gene for cell proliferation, this gene is not going to make any protein. Okay, there's not going to be any protein. It gives it a tight hug, no protein. Okay, no protein for cell cycle. So this is also a tumor suppressor. So your favorite ones are P53 and RB and P21. Okay. And this is an interesting one. The human papilloma virus, right? The ones that give rise to just warts, simple wart that people have on their skin, or cervical cancer, as you know it. So what the papillomavirus will do is kind of like it will go. So you look here how does the HPV increase the cell division? Well, it will do it because it will go and bind to the P53. So the P53 has, you know, P53 and this viral protein called, called as a E6 protein, E6 protein from the papillomavirus. So this E6 protein, this viral E6 protein will go and bind to P53, and then P53 will just not be there. It'll just be destroyed. So the whole thing will then be destroyed, and so P53 is absent. So it's almost like the human papilloma virus will simply take away your P53. And in case of these malignant tumors, there's a viral protein called as E7, and again, it will bind to the other proteins that help to make other proteins, and so it will bind that and it will prevent it. So, long story short, P53, is destroyed, and E7 protein binds to, RB. This thing is RB here, and so it just these viral proteins, they just bind to the tumor suppressors. Okay, it'll just bind to the tumor suppressors, and the tumor suppressors cannot function, or the tumor suppressors are degraded. So bad story both sides. So now let's talk about how these cancer cells metastasize. So those are all tumor suppressors now. So we just talked about those, and those are the basic names of the proteins. Let's see if I have them here. Anyway, I didn't put in the names here again, but uh, this is your ECM. The cancer cell is making MMPs. Remember, it is making MMPs to chop away all of these ECM molecules. Then you just learned about E-cadherin, its changes. Then you also learned about cytokeratin, all right, and N-cadherin and vimentin. And so these proteins will be either made and they change the cell. It's the proteins that change the cell. It's not the cells change and then make the protein. Okay, so the cancer cells manipulate them. So question for you: two cancer cells are taken from different areas within the solid tumor, and they're analyzed, which is false. Okay, so uh, just to so it's possible that the two cancer cells contain identical numbers of mutations. Well, let's see. Is that true? Um, that can be, right? But is it possible? It's possible that the two cancer cells do not share any of the same mutations. Um, then the fact is that these cancer cells, two cancer cells, this is false, that they will share some of the mutations of the same cancer critical genes, because we only have a couple. So those tumor suppressors typically, they do share. I mean, they will be common. So what did we learn here? We just learned that there are I didn't walk through the names here because we just did this today, but that you need specific genes for metastasis, you need specific genes for cell cycle. That's what we learned, right? Okay, so the angiogenesis, these tumor cells, we just learned that they will create their blood vessel or they will go to a place that has the blood vessel. And lung cancer will make its own blood vessels, you just saw in the video, and others will go to places where they can stay with them. So, so for angiogenesis, the molecular biology is that the cancer cells secrete vascular endothelial growth factor. Vascular endothelial, I hope you're taking notes, growth factor. So it's a growth factor, but it's for vascularization, and it works. It is made by the it is typically made by the endothelial cells, but in this case, the cancer cells are also making it, and so they tell the nearby blood vessel to make more branches of blood vessels. Okay, um, so that's how they do it, and we just saw how that happens. So more branching. And next is, the tumor microenvironment, the immune cells. The tumor microenvironment. How do the cancer cells take the help? From here, the cancer cells took the help of blood vessels, and here the cancer cells are going to take the help of the immune cells. And so this is don't look, be worried about this big picture here. So this is a cell. This is called as a T lymphocyte. It's a lymphocyte. Lymphocyte's job is to find anything foreign and throw it away or kill it. And this is the T cell nucleus. That's okay. That's the nucleus here. And this is your tumor cell. So this here is your tumor cell. Okay, this is your these are your tumor cells here. And these tumor cells, they express these tumor cells, the T cells and the tumor cells, they can express these specific proteins. They can express these specific receptors, or they can specific proteins. So what's a receptor? See this? This is a receptor, PD-1 receptor. It receives. How is it a receptor? If there is a signal, then the signal will just come here. It will receive it. Okay? So that's what a receptor is. It will just receive. So it's PD-1. So PD-1 receptor, and the other one on this side, take a look here. This is the CTLA receptor. And so this is your T cell. It's called as a T helper cell. So this is a T helper cell here. And these cells typically, their job is to go play tag, touch somebody, and that bad cell that it touches should normally die. Okay, so the cancer cell will manipulate this PD-1 and CTLA-4. So the cancer cells will inactivate the immune cells. So here's your cancer tumor cell or your melanoma cell. And so in the next slide, we show you, okay, you don't have to read all of this, but in the next slide, we show you that this uh, this PDL, okay, this PDL will go and it will just bind to PD-1 and it'll just stop it from, from doing any function. It'll neutralize it, okay? And same way CTLA-4, all right? And so in specific tumor cells, okay, they function in this way. So here they will bind to this receptor and it'll inactivate it. And so it just kind of like neutralizes it. And so there is, so that, sorry, this is normally what would happen. Okay, so these cancer cells the cancer cells will make these, okay, and make these PDL1 and CD860, and so then they can just go and play tag and then inactivate. These receptors are no more active. These receptors just don't work anymore. So it neutralize it, okay. So they kind of like neutralize it, and just say, okay, there's no function here. There's nothing to do. So this could have been otherwise active, but now it has kind of like no function. So the T cells just don't know what to do. And so in totality, you have your tumor environment. In totality, you have angiogenesis, and we looked at those in total. So at this point, I just want to share with you a video. Let's see if I can find the video. Um, are you looking at my right screen? Are you still on the cell cycle video, on the cell cycle slide, everyone?
Yes, ma'am. It is showing the cell slash cell.
No. Are you looking at the angiogenesis? Which slide are you looking at right now?
Angiogenesis, example one, angiogenesis.
Okay. All right. I don't understand why this is happening. I'm not, in my end, I'm seeing that. Oh. Okay, so let me just quickly just get out of here, and I want to show you a video. Oops. Okay, how did that happen? So I want to show you a video very quickly. And this is for cell cycle. And so that cell cycle is kind of like a big, but I didn't want to stop. So I wanted to, now want to show you this one now. And let me see.
So I think if any kind of question, please.
Yeah, just give me 1 second while I just do this. And this is a plain YouTube video, nothing fancy.
As a single cell, to grow, develop, and repair tissue. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day, our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into 3 distinct phases. During G1 or gap1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S or synthesis phase, the DNA replicates. Finally, during G2 or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as 2 sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
So
This familiar gelatin dessert actually.
So for the most part, it's the beauty of the cell cycles. So this class was really more for cell cycle and tumor suppressors. That's the molecular biology. That this is so well understood and so well studied that there for cytology, etc., there's nothing much to do. This is just well known. Okay, extremely well known. So so the fact is that things are just done, that understood so well, that these are used for when a genetic disease has been done. Let's say that there's a Down syndrome. There could be a Down syndrome baby. Then the fetus, actually before birth at a certain time, the cells can be checked for the fetus for I don't know, maybe around, I can't remember exactly how many weeks in, but it can be checked for chromosomal aberrations, etc., because of something that went wrong in the DNA replication or the spindle formation, the separation of the chromosomes. So this is kind of like really so well understood that those are already there. There's nothing much in terms of just of the whatever I said basic here to pick up for cytology, but they're always being done. So this is a basic concept for you to pick up from here and to look up for those that we don't understand. So with that, I would like to end today's talk, and I would be happy to take questions. I don't know how much time I took. So thank you, Dr.
And,
Ben, and
one
yeah.
question that is, I think, one question is raised by Parvati Madam. Parvati Dasgupta.
Yes,
Madam, are you in the link now?
I see her here.
Yeah. She has raised a question.
I can see her.
So I think Parvati Madam, any question, please? Okay. So we are in the verge of the ending of this session, and if any kind of question, please. And tomorrow in 10 o'clock, the next session, that is the Professor Nath's session, clinical, preclinical aspects, therapies, traditional and novel. This is the title. So tomorrow we will meet again with Madam at 10 o'clock in 30th October, the next day. So if any kind of question, again, Devushmita, any question? Devushmita?
Hello. Yes, sir.
Oh, Parvati
Hello.
Madam, please ask.
Actually, I have a question to Niharika Madam. Good afternoon, Madam. So, ma'am, my question is, you told that Lynch syndrome, it is a predisposition of any type of the cancer. So, I have to know about this more. That is, any type of the cancer. This is the starting point of any type of the cancer. There are different types of the cancers are there. So, Lynch syndrome this is the starting point.
No, it's
I have
for
to
colorectal cancer. For
Lynch syndrome. Okay.
yeah, so for not for any type, for some types, this is the like it's not for anything and everything, but yes, it can lead to typically to colorectal cancer. Let me just see if I can
Oh yes. So that means Lynch syndrome, it is totally linked with the colorectal cancer.
yeah, so let me just bring up this one. Thank you for raising that question.
Okay. Okay. Thank you, madam. Thank you very much.
Yes.
Okay.
Dr. Bhomik, are you here? Let me just check that topic. Oh yeah, clinical and preclinical therapies.
Yeah, for tomorrow.
Traditional and novel.
So thank you to all the participants. So our today's next session, I think we will start to 2:40 for a 40 minutes after break. We will call everyone. So I think today is the end of this session, and I think a lot of that is new conceptual biological cell biology thing Madam has already given a lot, so you can embed new ideas in computer science and engineering and how to use in our programming segment. So may be helpful for all of you. So thank you, madam, for this session. So
I hope they can build on it. I'm sure that they will be able to build on it, and there is nothing that is by itself, you know. It's so and you will be able to understand other papers if based on what we said so far.
Sure, and we are that is, we will send all the materials also, what we have done and madam has been done, all the slides also. So no issue. So thank you again, and Pooja, please formally end this session. Thank you, madam.
Thank you so much, madam, for the lecture for today. We will meet tomorrow at 10 a.m. So, after 40 minutes, we will start our next session at Google Meet. So, participant be there. Thank you, ma'am.
Thank you. Enjoy your next session. I will see you tomorrow. Bye now.
Recording stopped.
Bye, everyone. Thank you so much for being here.
Thank you, ma'am.
Bye, ma'am. Thank you, ma'am.
Thank you, ma'am.